Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in today's video I will be telling you about photosynthesis. Photosynthesis is chapter 13 of the A-Level Biology syllabus. So we are making progress very slowly but surely, and I hope that you have found many of the videos I've made very helpful. I've seen some of the requests for past papers to be analyzed, so I have done a random selection of some questions from past papers, and I will be doing a video on that very soon. So just stay tuned and turn on your notifications so that you can know when I post that video. But for today, I want to zoom in on photosynthesis. Now, I know that this is a chapter that many students find challenging. As a matter of fact, what I've noticed with many biology students is that they tend to find things that have to do with plants very unattractive for some reason. They just seem like, oh, it's not as interesting as when you're discussing something that happens in the human body. But I want you to know that photosynthesis is not difficult. It's really easy. And the purpose of this video is to demystify it for you. So like I said, my videos are mostly for revision, but they can also help you as pre-exposure. So you can watch the video before you go to the class, and you would have had some sort of foundational understanding. So when you get to class, you can ask lots and lots of questions. I love that when my students do that. So I always encourage them to watch videos before they come to the class so that we can have more of a conversation. So I hope that you can also see the videos in the same way. I have split this chapter into two videos. So this is the first of two. And in this particular video, I'm going to be telling you about the process of photosynthesis, the structure of the chloroplast, and just telling you about all the things that happen during photosynthesis. It might be a bit long, maybe 25 minutes, but the point is at the end of it, you will have a very good understanding of what happens during photosynthesis. So let's get into it, shall we? So starting off with the structure of chloroplasts. When we started this biology syllabus from the very first chapter, we started with the different organelles that you would find in a typical animal or plant cell. And we said that in the animal cell, we have the mitochondria, and in the plant cells, we have chloroplasts. And what you will learn, or what you will see about chloroplasts and mitochondria, is that they both have double membranes. So they're double membrane organelles, and they're believed to have been existing as independent organisms on their own before they were engulfed by eukaryotic cells. And that process of them being engulfed is called the endosymbiont theory. So if you haven't read about that, please make sure you just do a quick check or a quick scan in your book to get that. So if you look at a chloroplast under the microscope, you will find that they look like biconvex discs. They're very small in size, about 3 to 10 micrometers in diameter. And inside of them, which is where we're really interested, inside of the chloroplast, you will find these arrangements here. I like to think of them as coins just stacked on top of each other. And these are called granum. When you have a stack like this, it's called granum. When you have many stacks, they're called grana. That's the plural of granum. So I'm just going to use my red annotation tool here to sort of spell that out as gently as I can. Good. Okay, so many of these granum would be called grana, and each granum is made up of a stack of thylakoids. The thylakoids are the site of light-dependent reactions of photosynthesis. So you've probably heard before, if you have some biology foundation, that photosynthesis happens in two stages. You have the light reactions and the dark reactions. Well, the dark reactions are not exactly dark, but I'll explain that later. What this means basically is that you have a stage of photosynthesis that is highly dependent on the presence of light, and you have the second stage that is independent of the presence of light, which means that even if there's light or there's no light, that stage will proceed as it should. So we'll zoom into the thylakoid on a different slide, but I want you to know that that is where the light reactions happen. You also have in here what we call the stroma. And the stroma is like the cytoplasm of the chloroplast, and it's where you have the light-independent reactions of photosynthesis. So I want you to just bear that in mind, that the light-dependent reactions happen in the thylakoids and the light-independent reactions happen in the stroma. Now, when you zoom into a thylakoid, this is typically what you would find. A thylakoid has what we call a photosystem. And what a photosystem simply does is that it helps with the harvesting of light so that the photosynthetic reactions can happen. So if you look in here, for example, again, using my annotation tool, you can see that there are these little rectangles that are inside the funnel. So this is like a funnel arrangement. And what these rectangles do is that they simply trap light. So when sunlight shines on the photosystem, they trap the light and they pass the light down to each other until it gets to what we call a primary pigment. The primary pigment is usually something like chlorophyll A or chlorophyll B, and I'm sure you've heard of chlorophyll. It is what gives plants the green color, it is said. Chlorophyll also is very important in the process of photosynthesis, as you will see as we go along. We also have what we call the accessory pigment. So this is usually the primary pigment at the bottom of the funnel, and we have the accessory pigments that trap the light from the surface and pass it down to the primary pigment. So this is what happens in a thylakoid when you zoom in, and this is why it is the site for the light dependent stage of photosynthesis, because it simply allows for the harvesting of light. It is structured in a way that it harvests light and allows the primary pigment to partake in photosynthesis. The stroma, like I said, is a colorless fluid. It's the site for the light-independent reactions. It's colorless. It surrounds the granum or the grana. It also has enzymes, and these enzymes help with the light-independent stage of photosynthesis, which is also called the Calvin cycle. So you'll see the cycle once we get to the end of the slide. It's the very last slide. The Calvin cycle is what the light-independent stage is called. In the stroma, you would also find things that are typical of prokaryotic cells, because it is believed that the chloroplast used to be a prokaryote before it was engulfed by the eukaryotic cell. So you would find things like the 70S ribosomes, you would find DNA, secular DNA, you would find some lipids and starch grains, and all of these other things that you typically find in a prokaryotic cell. So now let's get into photosynthesis, shall we? This is the reaction for photosynthesis, and something that I ask students to notice immediately is that photosynthesis looks like the opposite of respiration. So if you remember when we were discussing respiration in detail, I said that if you want to get the reaction for respiration, it's basically glucose, which is that, plus oxygen, and then you make CO2 and water as your product. In photosynthesis, it is the reverse. You take CO2 with water, and then you make glucose and oxygen. So if you bear that in mind, it just makes it very easy for you to be able to remember. Light is often used in this process, like it said, photosynthesis. Photo meaning light, synthesis making. So light is used to split water into hydrogen and oxygen, and the oxygen is given off. But I'll show you how that happens when we discuss the process of phosphorylation. Also, this lends to the fact that when you think about photosynthesis, we always say that plants take CO2 from the environment and they release oxygen, while we take oxygen from the environment and we release CO2. And that just tells you why it is very important for us to exist in a balance with the plants around us, because we are simply contributing to the atmosphere in different ways and very dependent on each other. Now, like I said earlier on the previous slide, you have two types of photosynthetic pigments. You can have your primary pigments and you also have your accessory pigments. Accessory pigments are things like your carotenes that give carrots their yellow color, and your primary pigments would typically be your chlorophylls. You also have two different photosystems in photosynthesis. They're called photosystem 1 and photosystem 2. I wouldn't stress too much about that, but photosystem 1 is typically called P680, while photosystem 2 is often called P700. You also have what I explained earlier, that the accessory pigments would absorb light and pass that light energy down to the primary pigments, and the primary pigments would then carry out some excitation that results in the production of ATP and also the production of glucose as we go along. So now let's look at the light-dependent stage of photosynthesis. In the light-dependent stage, we have what we call the splitting of water. The splitting of water by light is referred to as photolysis, which is the word that I am underlining here. And just from the word photolysis, you know that it's using light to split something. And water is then used to produce hydrogen ions and electrons that are then passed around in some sort of electron transport chain and proton transport chain that will then result in the production of NADPH and ATP. To say that in a different way so that it just doesn't sound confusing, the point of the light-dependent stage of photosynthesis is to make NADPH and ATP. These two products are passed on to the light-independent stage where they are then used to produce the sugar that you need to make from the plant. So something to also bear in mind here, when we discussed respiration, we often said that in respiration we make NADH. And this NADH is passed to the last stage of respiration, which is oxidative phosphorylation, and that is how we make our ATP from the hydrogen that is removed from NADH. In photosynthesis, that is not NADH, it's NADPH. It's also like NADH in the sense that it is an electron carrier, and the easiest way to remember which one belongs where is to always know that the one with photosynthesis has a P in it, P for photosynthesis, so that would help you remember it better. So in the light-dependent stage, there are two key things that happen. You have phosphorylation and photolysis. I explained in the previous slides that photolysis is the splitting of water in the presence of light, and you'll see how that fits in very soon. But phosphorylation, what exactly is that? Phosphorylation is the addition of a phosphate, which means that what phosphorylation is referring to, it is the production of ATP by adding a phosphate group to ADP. And we have two different types of phosphorylation. You have either cyclic phosphorylation or non-cyclic phosphorylation. So in cyclic phosphorylation, this is typically what happens. The light comes from the sun and it would shine on the thylakoid. You know that inside the thylakoid we have this funnel arrangement that comes with different different pigments rather, that are arranged in what we call the light harvesting clusters. This is typically a photosystem. And in cyclic phosphorylation, this is photosystem 1, which is also referred to as P680. Once the light hits the pigments, these are the accessory pigments, the circles that I have included here, they would harvest that light from the sun and they would pass it down all the way to the primary pigment. Once the light reaches the primary pigment will then release an electron. So that electron is basically excited, and we call that a photoactivation. The electron is released, and once it gets to the top, it's released and captured rather by an electron acceptor. The electron acceptor will then pass the electron to an electron carrier. The electron carrier will then pass the electron through an electron transport chain until it goes back to the photosystem. The point of all of this is that as electrons travel, they release energy. And what that energy is then used for is to couple ADP and phosphate together in order to make ATP. Now, I know this can be a bit frustrating because it is so technical, well, not too technical, but it just seems like very detailed chemistry stuff. But bear in mind that just by summarizing it the way that I have done, you would be able to make it through the exams. So let's think back again. I'm just going to undo the animation so that we can sort of go back if we can. For some reason, my animations are stuck, so let's just go on. But I'll use my annotation tool here. So we start here where the light shines on the thylakoid. Inside the thylakoid, we know that we have what we call a photosystem. And in that photosystem, we have accessory pigments such as these ones that help to harvest light. And as they harvest the light, they pass the light down to the primary pigment. So I'm just going to call this primary pigment 1, okay? There's no space to write 1, okay, primary pigment 1. So once the light hits primary pigment 1 will release an electron. That electron is excited and passed on to an electron acceptor here at the top. That is called photoactivation. The electron acceptor will then pass the electron to an electron carrier, which will then take it through an electron transport chain. And as it does that, the electron provides the energy, just by its movement, provides the energy that is able to then allow the coupling of ADP and phosphate in order to make ATP. The reason why this is called cyclic phosphorylation is that the electron that is excited is passed back down to the photosystem. It doesn't go anywhere else. It goes to the electron carrier, releases energy, and comes back down to the photosystem. That is why we call this cyclic phosphorylation. In non-cyclic phosphorylation, things are a little bit more complicated and also very interesting, but not that complicated that you can't get it. Oh, I was wrong. Photosystem 2 is P680 and photosystem 1 is P700. That was a mistake I made on the previous slide. Sorry, I mixed that up. So please bear in mind that photosystem 2 is P680, photosystem 1 is P700. Not that you're often asked questions about this, but it's just important to get the right information out there. So now let's look at what happens in non-cyclic phosphorylation. First things first is I want you to notice over here what is happening with water. All right, water is coming in here and you can see that it is being split. If you follow the arrow over here, it is being split into oxygen and hydrogen. I hope you can see that. So let's just go through this. I just wanted you to notice that is photolysis happening. And then you also have phosphorylation, but in a non-cyclic style. So what happens in this case is that light will come in. So I'm just going to draw something that looks like the sun over here. Perhaps I should use a yellow marker for that, you know, just because we always look at the sun as something that's yellow. So there we go. So light comes in here and it hits this photosystem. Okay, this is photosystem 2. The light hits photosystem 2 and photosystem 2 would release an electron the same way it happens in cyclic phosphorylation. It releases an electron, and what then happens is that electron goes through an electron transport chain. And obviously, as it goes through the electron transport chain, you already know that provides energy for ADP and phosphate to be bound together so that they can make ATP. So they basically take the energy and then make ATP. So that then happens, but look at what happens here. The electron that travels from P680, which is photosystem 2, will then go to P700, which is photosystem 1. And at photosystem 1, what happens here also is that when light hits it, it also has an excited electron. So let's think about it this way. Let me just re-explain it from the beginning. I just noticed here now that we actually have light that's just being hit here. So here's our light, okay? Let me go back to my red marker. So here's light, and light is hitting our photosystem. All right, I'm just erasing some of the other stuff that's not necessary. So here's our light here, and it's hitting the photosystem. Okay, so this photosystem releases an electron as a result of the light hitting it, which means that there's photoactivation because the electron gets excited. But it doesn't stop there. On the second photosystem, which is photosystem 1, P700, light also hits that photosystem and it also releases an electron. Now look at what happens here. The electron that is released from P680 will travel through an electron transport chain and replace the electron from P700 that has been excited. The electron that was excited from P700 is then donated to NADP in order to make NADPH. All right, so think about it. The electron that goes from here, P680, if you follow the arrows, this makes it a lot easier. The electron that's excited from P680 goes through an electron transport chain and replaces what was lost from P700. Remember that P700 was also hit by light, so it has also released an electron, and that electron is used in the formation of NADPH. The question students usually ask me is that, okay, then what happens to P680 because it is missing an electron? That electron is replaced by water. So when you have photolysis, you release electrons and protons, and so the electrons will then replace whatever has been lost from P680, if that makes sense. And just by this movement of electrons that we have here in a continuous fashion, we have energy in the form of chemiosmosis. So I hope you remember this from respiration, chemiosmosis. that is then used to couple ADP and phosphate in order to make ATP. So I'm just spelling chemiosmosis here, painfully careful about writing without any pens, but just using annotation tools. So this is called chemiosmosis. So it provides the energy for ADP to then bind with phosphate. So we have ADP plus phosphate, and that is what makes ATP. And the ATP and NADPH that we make from this phase of the reaction, this is NADPH over here, the ATP and NADPH that we make is then transferred to the light independent stage. In this case over here, the oxygen that is given off, that is produced from photolysis, is given off as gas, and that is the oxygen that we take in from the environment, while the hydrogen ions obviously are used also in making NADPH. This doesn't show that very clearly, but that is typically what happens. So that is the light dependent stage. As you can see, nothing much happens there. It's really just an electron transport chain where electrons move around and NADPH is produced as well as ATP. Those two things, NADPH and ATP, then move to the next stage, which is the light independent stage. The light independent stage is also called the Calvin cycle. And this is where things might be a bit complicated for students, but typically it's not. First of all, I'll tell you something. You are not expected to know the molecules or the number of molecules involved in the Calvin cycle. As a matter of fact, I think it says in the syllabus for 2018 to 2020 that you don't have to know the biochemistry. So you don't have to know the number of molecules or the arrangements, but you do have to know the names of certain important intermediates, and that is what I'm going to show you with this slide. So this image that I got from lumenlearning.com, I use it in the classroom to explain the stage of photosynthesis, so I hope that it is also easy for you to follow. So think about it. We've made ATP and we've also made NADPH. Then we come to the Calvin cycle. In the Calvin cycle, CO2, which we know is very important in photosynthesis, comes into the picture. CO2 enters the Calvin cycle and it combines with a compound called RuBP. The full name for RuBP is Ribulose bisphosphate. I will put that in the description so that you're able to follow, just be able to get the spelling. So most of the things that I'm unable to say in detail or that might be confusing, I'm just going to put in the description so you can easily just check that. So CO2 comes in, it combines with RuBP. If you look here at this image, RuBP has 5 carbons. So there's 1, there's 2, there's 3, there's 4, and there's 5. So if you add this 5 carbon compound with a 1 carbon compound, you're going to make a 6 carbon compound. That 6 carbon compound is highly unstable. So it's often not shown in the Calvin cycle. It's highly unstable and it immediately splits into 2 molecules of a 3 carbon compound. Again, ignore the number of molecules here. I'm dealing with this as if I'm working with just 1 molecule. That is why it looks the way it does. So don't be afraid if you hear me say 1 molecule, but you're looking at it and you're saying this is 3. So if I'm dealing with it like just 1 molecule of CO2 with 1 molecule of RuBP would make 1 molecule of 6 carbon compound, which then splits into 2 molecules of a 3 carbon compound. Those 2 molecules of a 3 carbon compound then undergo a series of reactions. And as you can see here, we make ATP over here. We use ATP rather over here. So ATP is used. Wherever you see ATP going to ADP, that means ATP has been used. And this is the ATP we made from the light dependent stage of photosynthesis. And NADPH is also used. As you can see there, it goes from NADPH to NADP, which means the hydrogen ions are also used. And that goes on through there. So we have a reduction reaction of this intermediate here. That reduction reaction results in the formation of this molecule called glycerate 3-phosphate. I'm going to put that again in the slides, so don't stress too much about it. So from 1 molecule of RuBP combined with CO2, we've made these intermediates and we end up with glycerate phosphate. 1/6 of the glycerate 3-phosphate molecule is converted to half a molecule of glucose, or is converted to glucose. You don't have to worry about the number of molecules yet again. It's converted to glucose, while the remaining 5/6 molecules would be taken through a series of reactions in order to remake RuBP. So if you think about it, just pay attention here. There are different stages, 3 key stages in the Calvin cycle. The first stage is what we call the carbon fixation, which means CO2 comes in, it combines with RuBP, and that is what we call the carbon fixation reaction. Something else to bear in mind here is that the enzyme that catalyzes the formation of the 6-carbon intermediate formed after CO2 and RuBP combine is called Rubisco. So Rubisco catalyzes the reaction between RuBP and CO2 over there. Right, once that is added together, once those two add together and they form a 6-carbon intermediate, the intermediate splits into 2 molecules of a 3-carbon compound, which is this over here. We then go through a series of reactions where you use ATP and NADPH from the light-dependent reaction, and we form glycerate 3-phosphate. From glycerate 3-phosphate, 1/6 of that is used to make glucose. So we take 1/6 of that and we use glucose, and the other 6th is then used to, the other 5th rather, the 5th, well, the 5 out of 6 is used to make RuBP. So we're basically saying that from the 6 carbons, from what's it called now, from the 3 carbons, if we combine the 3 carbons, what's called together, into 2 molecules, we would make a 6-carbon sugar, which would be glucose. But then the other carbons that have been made, the other 3 carbon compounds that have been made, are used to regenerate RuBP. Don't be too stressed if this is a little bit confusing. Just always think of it in terms of the steps. You have carbon fixation, where CO2 combines with RuBP, catalyzed by Rubisco to make a 6-carbon intermediate that is highly unstable, and that then splits into 2 molecules of a 3-carbon compound. ATP and NADPH from the light-dependent reactions are used to reduce that 3-carbon compound to glycerate 3-phosphate, and from glycerate 3-phosphate we use some of these molecules to make glucose, while the rest of it is used to regenerate RuBP, and so the cycle continues in that way. If this has been confusing, please just post questions in the chat. I know that the Calvin cycle can stymie students a lot, so I'll totally understand. But post any questions that you have in the chat, anything you'd like me to just re-explain, perhaps in words, that I can type to you, and I would be happy to answer them. This is the end of this very first part of photosynthesis, and I will continue in the next part, where I tell you a bit about C4 plants and then about the factors that affect photosynthesis. Until then, have a good time, and thank you so much for watching.